So I'm Paul Orchard. I have the opportunity to introduce Mark today. But before I get to that, I just wanted to spend a couple of minutes talking about Bill Krivett. So I know a lot of people, especially the younger people, never had an opportunity to meet Bill. And we lost Bill about 10 years ago. But Bill was kind of a fixture at the university for over 50 years. He was one of the fundamental instigators of the Children's Cancer Study Group, or research group, that evolved into a children's oncology group. So he played an important role in that. He published some important papers in coagulation and hematology and oncology. But what Bill was really known for, and he actually was the chair of the department for 7 years or so. But the thing that Bill was really remembered for is his role in development of transplantation for metabolic disease. And in regards to that, he was, under his leadership, the first patient in the country was transplanted here back in the early 80s, and this is the result of that paper. And he also was important in the leukodystrophies, and Mark's going to be talking about transplantation and gene therapy for Globoid Cell Leukodystrophy today. So, again, Bill was internationally recognized as a leader and a pioneer in the terms of transplantation for these various diseases. So, and we have the pleasure of having Mark Sands with us today. So Mark originally is from Rochester and did some of his training in New York, and then, so I saw you were actually Jackson Labs in Bar Harbor for a few years. I'm sure there's some good stories related to that. And then did another postdoc at University of Pennsylvania, and since that time spent at Washington University in St. Louis. So Mark is an incredibly bright guy, very well respected in terms of treatment for lysosomal storage diseases and the use of gene therapy. It's got over 100 publications in top-notch journals describing approaches for gene therapy for these disorders. So, without any more ado, we'll let Mark get started.
Can everybody hear me? Way back in the back? Okay. If not, just yell and I will do my best here. Okay, so before I get started, I'd like to thank Paul for inviting me here to give the annual Krivett Lecture. I was one of those people that never had the opportunity to work directly with Bill, but I certainly met him at several meetings, and it's certainly an honor for me to give this lecture that's named after one of the pioneers for using bone marrow transplant for lysosomal storage diseases. So thank you, Paul. I appreciate it. So what I'll do today is talk about some of the recent data, both published and unpublished, that we've been working on trying to develop an effective therapeutic strategy for Krabbe disease. Let me see if I can work this. There we go. So for those of you who don't eat, sleep, breathe these very rare diseases, I'll give a quick overview of Krabbe disease, which is also called Globoid Cell Leukodystrophy. Krabbe disease is a lysosomal storage disease by the fact that the lysosomal enzyme galactocerebrosidase is deficient in Krabbe disease. Now, this disease is inherited as an autosomal recessive disease. It's profoundly neurodegenerative, and the function of GAL-C, which I'll refer to for the rest of the talk, is to remove the galactose from galactosylated lipids. Now, the major lipid that GAL-C works on is galactosylceramide, which of course is one of the major lipids, one of the major myelin lipids. But there's also a rare species of galactosylated lipid called galactosylsphingosine, or what I'll refer to as psychosine for the rest of the talk. And it just so happens that psychosine is a highly cytotoxic galactosylated lipid. If you add this lipid onto virtually any cell type, it will kill it. It's a very nasty little lipid. But since cycosine is produced primarily in oligodendrocytes, the first cell type that dies in Krabbe disease, in fact, are oligodendrocytes. And that leads to this demyelinating, or probably more appropriately, a dysmyelinating disease. It's a progressive disease, and one of the hallmarks of Krabbe disease is this progressive and very profound neuroinflammation. It affects both the central and peripheral nervous system, and clinically, these kids begin to present, at least in the infantile form of the disease, they present very early in life with motor deficits, progressive cognitive decline, and intractable seizures. And untreated, at least in the infantile form of the disease, these children typically die at 2 to 4 years of age. It's a very aggressive, very nasty disease. Now, we happen to be very lucky in the field that there is a phenotypically accurate model of Krabbe disease, and that's called the Twitcher mouse. The Twitcher mouse was a spontaneously arising mutation that was originally discovered at the Jackson Laboratory in the late 1970s and was published in 1980. Like the human disease, it's completely deficient in GAL-C activity. It has a demyelinating, again, or dysmyelinating phenotype, affects both the central and peripheral nervous systems. And I think this really models the infantile form of the disease quite well. It's a very rapidly progressing disease. These mice have a median lifespan of only 36 to 40 days of age. And here's a picture of a 36-day-old Twitcher mouse right here. And because this is not a video, you can't really appreciate. This mouse would have had a very severe tremor, and it develops hind limb ataxia to the point where between 36 and 40 days, its hind limbs are completely paralyzed. Again, it has a severe tremor. This initially begins to manifest itself right around weaning. The hind limb ataxia then is a later phenotype. And like the human disease, there's progressive and profound neuroinflammation associated with the murine model. So again, this is really a very useful and accurate phenocopy of the human disease. And it's been used extensively by a number of groups. Now, histologically, this shows the hallmark of Krabbe disease. This is a section of cerebellum that's stained with Luxol fast blue and periodic acid shift. This is from a mouse, from a wild-type mouse. And you see this very nice looking white matter track through the cerebellum. You see all this blue staining. That's the Luxol fast blue staining the myelin. And it's very nicely organized. It's a very nice looking white matter track. And you see a few cells that stain positive with PAS. Those are likely oligodendrocytes through the white matter tracks. This is a white matter tract through the cerebellum of a Twitcher mouse. Okay? First thing you should notice is there is less blue staining, so a decrease in myelin. But the other thing that you should see are these very large PAS positive cells that are multinucleated. And that really gives the disease its name. These are referred to as globoid cells. And the white matter tracts are just littered with these, again, multinucleated PAS positive cells. And again, that gives the disease its formal name. And I apologize for the fuzzy picture, but it's pretty easy to see. Again, there's this profound neuroinflammatory component of Krabbe disease. Here's sections of striatum from a wild-type animal stained with CD68, which is upregulated on activated microglia and macrophages. You see lots of CD68 staining in a Twitcher mouse, and this is at 36 days, so towards the end of the animal's life. But we also see very severe and profound astrocytosis also in the striatum, and actually you can see it throughout the neuroaxis of these animals. So this is an example of the neuroinflammation, and this is also mirrored in the dysregulation of a number of cytokines and chemokines. This is just a relatively small survey of cytokines and chemokines, and you can see that a number of them are dysregulated, and in particular, keratinocyte chemoattractant factor, which for you folks that study human cytokines and chemokines, this is IL-8 in humans. And you can see that there's a lot of dysregulation, again, coincident with the progressive neuroinflammation. So since this is such a nasty disease, what is the effective treatment for this disease, or is there an effective treatment for this disease? The simple answer is no, but I'll walk you through very briefly a very superficial review of the history of treatment for Krabbe. So in 1984, Andy Yeager performed the first bone marrow transplant in the Twitcher mouse, again, which is the model of Krabbe disease. And as I mentioned before, the median lifespan of the Twitcher mouse is between 36 and 40 days of age. Well, following a bone marrow transplant that Dr. Yeager did, where he took 10-day-old Twitcher mice, so postnatal animals, about 10-day-old, still very young, gave them 900 rads of radiation and injected them with Gauss-positive donor cells. And given that scenario, so given lethal radiation at postnatal day 10, he could increase the lifespan to about 80 days. So he doubled the lifespan of these animals. We do bone marrow transplants in neonatal animals, which is a bit of a problem. Neonatal animals do not survive, or they do not tolerate the conditioning regimens very well. So we're forced to give sublethal conditioning, about 400 rads of radiation. We get low-level engraftment, about 20 to 30%, and we get a statistically significant increase in lifespan, but it's only to about 48 days, okay? And that's probably due to the low-level engraftment. Either way, bone marrow transplantation can have some clinical impact on the disease, but it's really very minor. Again, 80 days is not much of a lifespan when the normal lifespan of a mouse, laboratory mouse, is about 800 to 900 days, okay? Now, what's unfortunate is it seems we get a similar response in children. In other words, from all the data that are out there, it looks like bone marrow transplantation can have some benefit for Krabbe children, but it's really, at least in my opinion, moderate to minimal at best. The disease continues to progress. These kids have a very poor quality of life, but interestingly, that is currently the standard of care for Krabbe disease. So what's been done trying to develop additional or more effective therapies? So this is a very short list of a number of things that have been attempted in the Twitcher mouse, so in the murine model over the years. And of course, stem cell transplantation, anybody that works with their favorite stem cell has tried to transplant those cells into the Twitcher mouse. We already talked about hematopoietic stem cells, but neuronal stem cells, mesenchymal stem cells, oligodendrocyte progenitors. Everybody has tried to inject their favorite stem cell into these animals with essentially no response, okay? They've tried anti-inflammatories, antioxidants, substrate reduction therapy, dietary intervention, and I could have made this a very long list, but the take-home message is no single therapy is effective in Krabbe disease. Now, so you might get a day or two increase in lifespan, but really not effective. So we started studying the mouse model of Krabbe in the late 1990s, and partly because we worked with MPS 7 for many years, and we had just published a couple of what I think were relatively important papers using a gene therapy approach. And in the late 1990s, there had been very little gene therapy done in the Twitcher mouse model of Krabbe. And we showed that, at least in MPS 7, if you do an intravenous injection of an adeno-associated viral vector in neonatal MPS 7 mice, you can nearly completely correct the phenotype of the animal, but also their vision is corrected, their hearing, skeletal system, lifespan. It was really very effective. If we injected the AAV vector directly into the brain in neonatal animals, we could prevent the cognitive deficits associated with this disease. So we naively thought, we can probably do the same thing in Krabbe. It's a lysosomal storage disease. There's cross-correction. This should be pretty straightforward. So we tried that exact experiment. And in this particular experiment, we directly compared AAV2, which is the original AAV vector that was developed, and then an AAV vector pseudotyped with the AAV5 capsid, which at the time we started these experiments was the darling of the gene therapy world with respect to CNS gene therapy. So it was a very simple experiment, just injected the virus intracranially, very much like we did with MPS 7. And this is the only slide I'll show from that paper. Here's the lifespan of untreated Twitcher mice. Here's the lifespan of Twitcher mice injected with an AAV vector expressing GFP. Here's the lifespan of animals injected with AAV2, animals injected with AAV5. Both of these lifespans are significantly greater than untreated animals, and AAV5 is significantly better than AAV2. But again, if you look at the horizontal axis here, we really haven't done much. And again, this is consistent with what I said before, that no single therapy is really very effective in this disease. Okay? Even though with MPS 7 it was wonderful. So we took a step back and thought more about this disease. It is, like most of the lysosomal, like all of lysosomal storage diseases, it's a simple monogenic disease. But if you think about the disease itself, it has a very complicated clinical picture. There's, of course, the primary defect, but then there are all these other secondary pathogenic mechanisms that are initiated as well. So we thought, well, if single therapies won't work, maybe we need to target the primary defect along with some of these other secondary consequences. So the first experiment I'll talk about is where we did CNS-directed gene therapy, just like I showed in the previous slide, but we coupled that with bone marrow transplantation. Now, the rationale for this approach was that the gene therapy would target the primary genetic defect. And one thing that I didn't tell you about that first experiment we did was that if we do CNS-directed gene therapy with an AAV5 vector, we get about 3 to 5-fold higher than normal levels of enzyme in the brain. So supraphysiologic levels of enzyme. So, in fact, we know that this is attacking the primary genetic defect. It's supplying GAL-C activity. And the bone marrow transplant, at the time, it was believed that the bone marrow transplant, of course, through migration of donor-derived cells into the brain and then cross-correction, that the bone marrow transplant would actually decrease the level of cytosine. But there was also a growing body of literature at the time, suggesting at least, that bone marrow transplant could have an immunomodulatory effect. So we didn't exactly know what this was going to target, but it's the standard of care, and we know it's doing something in there. So the experimental design for this experiment was that we would identify the animals at birth, and at postnatal day 2, we'd genotype them. Since they have no phenotype at birth, we'd genotype them at postnatal day 2. At postnatal day 3, we would give them both an intracranial injection of virus, just like I described previously, but we also discovered that an intrathecal injection would also decrease the disease burden in the spinal cord and possibly even some of the peripheral nervous system. So intracranial and intrathecal injection of AAV. And on postnatal day 4, we'd expose them to 400 rads of radiation. Again, these animals will not tolerate any more radiation than that. And then give them an intravenous injection of bone marrow, donor-derived bone marrow cells. And these bone marrow cells are GAL-C positive, but they're also constitutively expressing GFP, so we can follow those cells later. Okay, and keep in mind, as I go through the data, with only 400 rads of radiation, these animals are only engrafting to about 20 to 30%. Okay, so we get stable mixed chimerism, and actually it's fairly low with respect to the donor-derived cells. So, of course, the first question we asked was, are we getting enzyme activity in the brains of these animals? Okay, and up here, here's the level of GAL-C activity in a wild-type animal. Here's the level of enzyme activity, or if you will, the lack of enzyme activity in an untreated Twitcher mouse. And this is where things began to get interesting. Following a bone marrow transplant only, there was no statistically significant increase in enzyme activity in the brains of those animals. Now, you'd get the odd animal where you'd get a little bit of activity, but in most of the animals, it was virtually undetectable. So right there, that sort of challenged one of the original dogmas about bone marrow transplant, that cross-corrective enzyme was actually mediating any sort of beneficial effect. Now, when we look at the GAL-C activity supplied by the AAV, though, so this is wild-type levels, and as you can see, there's higher than normal levels of enzyme activity whether it's AAV alone or AAV plus bone marrow transplantation. And you can see that histochemically. There's a histochemical stain for GALC activity, which deposits a blue stain. And you can see sort of low-level diffuse GALC activity throughout the spinal cord, cerebellum, and forebrain, a wild-type animal. No enzyme activity in a twitcher. But then you see a lot of enzyme activity here, and in particular in the ventricles, back here in the fourth ventricle as well. You see it around the spinal cord. Again, not surprisingly, we did intrathecal injections in these animals. But what it's harder to appreciate is you also see a fair number of GALC positive cells outside of the ventricles as well. There's actually quite a bit of enzyme activity there. Again, consistent with our quantitative assay. So the next question, of course, is we're supplying GALC activity. Are we decreasing cycosine? So are we decreasing the level of this toxic metabolite that accumulates? And when we look in the brain, here's the level of cycosine in an untreated wild-type animal. It's essentially undetectable, even using very sensitive mass spectrometry methodologies. Essentially undetectable. And here's the level in an untreated twitcher animal. There's a significant increase in cycosine. What was surprising to us, though, is when we looked at animals that received bone marrow transplant alone. I just showed you that there was essentially no GALC activity resulting from the transplant. And in fact, there's no reduction of cycosine either. In fact, there's a statistically significant increase in cycosine in animals that received a transplant alone. Now, I don't know why there's actually an increase following the transplant, but the important thing is the transplant does not decrease cycosine at all. So it supplies essentially no enzyme activity, no decrease in cycosine. In contrast, when you look at animals that were injected with the virus, either alone or in combination with bone marrow transplant, here's the level in an untreated twitcher animal, and you see this significant decrease in animals that were injected with either AAV or AAV plus bone marrow transplant. But you see that there's basically no difference in those two. So any reduction in cycosine that we see is coming from the gene therapy, not from the bone marrow transplant. And what I'd also like to point out at this stage is this is not completely normal. So there is still cycosine in there. We have not eliminated the accumulation of cycosine. Okay, so keep that in mind when I talk about some of the future studies. So we also looked at the sciatic nerve, the level of cycosine there. Since the peripheral nervous system is also affected, we use the sciatic nerve as a surrogate for the peripheral neuropathy. And you see virtually the exact same pattern. Here's the wild-type level of cycosine in the sciatic nerve, untreated twitcher, following a bone marrow transplant. There's no reduction in cycosine in the sciatic nerve. In contrast, any animal that was injected with AAV, again, either alone or with bone marrow transplant, there was a significant reduction in cycosine in the sciatic nerve. So then we wanted to ask more, if you will, sort of clinically relevant questions. So in collaboration with Victor Song at Washington University, we performed Dti measurements in these animals. And the quantification is down here, but these images really speak volumes, so we'll focus up here. This shows the axial diffusivity in the spinal cords of wild-type animals, mutant animals, animals receiving the combination, wild-type gene therapy alone, AAV alone, combination animals are untreated. And if you look at the axial diffusivity, which is an indirect measure of axonal health, you can see here in the ventrolateral white matter tracts and in the dorsal white matter tracts, the signal is much less than in the wild type. Again, suggesting that there's significant axonal damage in the untreated twitcher animal. And both of these treated animals, either AAV alone or combination, look much closer to normal than they do mutant. Similarly, when we look at radial diffusivity, which is an indirect measure of the degree of myelination, which of course will inhibit the diffusion of water, at least radially. Again, here's the wild type animal, and you see a significant increase in the signal in the twitcher animals, again suggesting or indicating that these animals have less myelin and there's more radial diffusion of water compared to the wild-type animal. And again, if you look at both of the treated animals, AAV or AAV plus bone marrow transplant, these animals look closer to normal than they do the twitcher. Okay, so we're getting some benefit that we can actually measure by MRI. So here's obviously probably one of the simplest outcome measures we can have, but also perhaps one of the most meaningful, and that's simply lifespan. If we look at the lifespan of these animals, here's the lifespan of untreated twitcher animals. Here's the lifespan of animals that receive bone marrow transplant alone. Again, you get anywhere from 5 to 10 day increase in lifespan. With AAV5, you can increase the lifespan from about 36 to 40 days to maybe about 50 to 55 days. So really very minimal response from either of those treatments alone. But this was what was interesting to us, the combination. When we treated animals with both bone marrow transplant and AAV, CNS-directed AAV, you saw this dramatic increase in lifespan. Now, we're still not terribly happy with this, because again, if you look at the lifespan, the median lifespan really is about at 120 days. Okay, and again, compared to 800 or 900 days for a normal mouse, this still isn't great. But what we were excited about was this interaction. Clearly this interaction, this increased lifespan, indicates that there's some synergy going on between the bone marrow transplant and AAV5. And perhaps if we could understand this synergy, perhaps we could exploit that to improve these therapies even further. So again, we were less excited about the magnitude of the response, but more excited about the synergy that we saw. So what is the mechanism of synergy? So the simple answer is, I'm not going to stand here and say I completely understand this, but I think we know at least one component of it, and that's this effect on neuroinflammation. Okay? So remember, one thing we can say at this point is that the bone marrow transplant is not mediating its effect through cross-correction or reduction of cytosine. We showed that earlier on. But when we look at the inflammatory cells in the brains of these animals, this was where the surprise came in. And it's really quite dramatic. If I go to the right thing here. So if you just focus on the contour plots here, the quantification is down here, but again, these contour plots, you can get the story from there. If we look at the infiltration of T cells, so CD8 and CD4 cells into the brains of these animals, you can see in a wild-type animal, there's very few CD8s and CD4s. There shouldn't be very many there. Interestingly, in the untreated twitcher animal, there's also very few CD8s and CD4s, suggesting that this neuroinflammatory response is really not a T-cell-driven response. Okay? What was surprising to us, given all the literature out there about AAV and how safe it was and how there wasn't an immune reaction against AAV, we saw that animals treated with AAV alone had a significant increase in CD8s and CD4s. Now, what I can't tell you is whether this is a response against the AAV capsid proteins or whether it's a response against the recombinant enzyme that's being made. We didn't do that experiment, so I can't tell you. But what was truly surprising to us was when we looked at the inflammatory cells following treatment with AAV plus bone marrow transplant. This CD4 and CD8 response is completely abrogated when you add bone marrow transplant to the CNS-directed gene therapy. This was a huge surprise to us. It was equally surprising when we looked at the number of activated microglia and macrophages in the brain. Again, in the wild-type animal, so what you're looking for is these quadrants up here. Very few activated microglia and macrophages in the brains of wild-type animals. You see this significant increase in untreated twitcher animals, which again is consistent with the nature of the disease, these globoid cells that infiltrate into the brain. This microglial macrophage response was exacerbated in animals treated with AAV alone, consistent with what we see with the T-cell response. But again, what was so shocking about this was when you add a bone marrow transplant to the gene therapy, you eliminated both the disease-associated and the gene therapy-associated microglial macrophage activation. It was truly shocking to us. So we think that at least in the context of the experiments that we performed and the way we performed them, The bone marrow transplantation is really, again, not supplying enzyme through cross-correction. It's not decreasing cycosine, but it is having this very significant immunomodulatory effect. And we think that's at least a major component of the synergy we see between these two therapies. So, I showed you the lifespan curve following treatment with CNS-directed gene therapy and bone marrow transplant. We got these animals to live to about 120 days, which again, we were excited about the synergy. We were excited to discover that the bone marrow transplant has an immunomodulatory effect, but still 120 days is not much, okay? So, being good Americans, bigger is better, we said, what the heck? Let's try an additional therapy. Let's target another pathogenic cascade here. So what we did is we did the exact same experiment that I just described, CNS-directed gene therapy. And these experiments up to this point are all being performed with AAV5. Okay, we have not changed the bone marrow transplant protocol or the gene therapy vector at this point. So AAV5, bone marrow transplantation, but we added a small molecule substrate reduction drug. Okay? And the rationale for this was the gene therapy would target the primary genetic defect and supply high levels of GALC activity. We now know that at least the way we do our transplants, the bone marrow transplant is having primarily an immunomodulatory effect. But the substrate reduction therapy, in principle, should reduce the accumulation of the substrate, so this the galactosyl sphingosine or cycosine. In principle, that's what it should do. And the small molecule drug we gave was L-cycloserine, which you can see right here. We also thought that since this was a small molecule drug, we may have better penetration into the CNS and perhaps a broader distribution into the CNS where we could potentially target those areas that the gene therapy wasn't targeting, or perhaps where we were getting subtherapeutic levels of enzyme activity. If you now decrease the synthesis of the toxic metabolite, those two may interact and get you over that therapeutic threshold. So that was the rationale. Experimental design was very similar to what I just described. Genotype the animals at postnatal day 2. Postnatal day 3, they get the intracranial and intrathecal injection of AAV5. Bone marrow transplant at postnatal day 4. And then starting at postnatal day 5, we would give 3 times a week subcutaneous injections of L-cycloserine at a dose of 50 mg per kg. And these animals, the animals that are still alive, are still getting this regimen, 3 times a week, substrate reduction. So we'll go through the same typical assays that we do. What about GALC activity? Again, here's wild type levels, here's the twitcher, and at 36 days, here is the triple treated levels of enzyme activity. Very much like what we saw before, not at all surprising. We're giving the same viral vector, okay? And one thing you can tell already is we have a long time point here, so that's sort of giving away the punchline. But you see this, what looks like a decrease in enzyme activity as these animals age. There's no statistically significant difference between these groups, but it would appear as though the enzyme activity is decreasing. I don't know if it is. We've only got an N of 2 out here. But even if it is decreasing, at this when these animals are terminal, which these 2 animals were actually out at around 350 days of age, they're still several-fold higher than normal animals. So we still have supraphysiologic levels of enzyme activity. What about cycosine? Same sort of story. Here's cycosine levels in a wild-type animal. Here's the untreated twitchers. And here's at 36 days, 160 days, and there's 2 terminal animals. You can see that the level of cycosine remains quite low. Again, it's not normal. There's still elevated cycosine, but it's significantly less. It's actually dramatically less than an untreated twitcher animal. Okay, and again, this persists for the life of the animal. What about the neuroinflammation? So the neuroinflammation, again, this is CD68 staining, so you're looking for activated microglia and macrophages. Here's an untreated twitcher, cortex, cerebellum, brainstem, wild compared to a wild-type animal. You can see at 36 days, consistent with what I showed before, there is a decrease and we've quantified this, it's a significant decrease in CD68 staining. There's also a significant decrease in GFAP staining, so astrocytosis is decreased as well. But unfortunately, as these animals age, the neuroinflammation seems to come back. And in fact, here at the terminal time points, you could make an argument, this is not statistically different from an untreated animal, but it's certainly pretty severe. Okay? What's odd, though, is that there's an inconsistency here with the neuroinflammation. And I will freely admit right now I'm not an immunologist, so I'm not going to pretend I understand this. But if you measure CD68 staining or GFAP staining, you can see the staining go up as these animals age. But interestingly, when you look at cytokines, KC, FGF, IL-10, MCP1, doesn't matter which one, you see that they're virtually normalized, or at least very close to normal, at an early time point for all of these cytokines and chemokines. But as they get older, even though the inflammation, as we measure it by immunohistochemistry, increases, the cytokine and chemokine levels remain very low. In fact, are nearly normal in all these animals at all these time points. I don't understand that. I mean, somebody who's smarter than me and who actually understands immunology, if anybody actually understands immunology, would have to explain this to me. So there is a inconsistency here, but this is quite encouraging, actually, that at least the cytokines and chemokines remain relatively low for the life of the animals. So here's one of the punchlines in this particular study. There's a whole bunch of curves in here. The green is wild type. Yellow is actually untreated twitcher, so there is actually a yellow line underneath there. The black line is animals treated with bone marrow transplant alone. Orange is L-cycloserine alone. Blue is AAV alone. Again, they have minimal to no effect. They don't have no effect. They have very minimal effect. And the red line is my favorite because it's one of the few chances I get to make up data. If you took the increase in lifespan from all three of these groups, you would get a lifespan, if they were additive in nature, this would be the lifespan of those animals. Clearly, that's not the lifespan. This is the actual lifespan. And again, by now targeting another pathway, so by inhibiting the enzyme that produces, or one of the enzymes that's responsible for producing cycosine, along with the other therapies, you get this initial boost in lifespan. And in fact, the median lifespan here is about 300 days. It's actually 298.5. We couldn't squeeze out that extra 2.5 days, or 1.5 days here. So a dramatic increase, again, from 130 days with bone marrow transplant and gene therapy alone to about 300 days when we target another mechanism. So what is the L-cycloserine doing in there? I mean, according to all the literature, I mean, L-cycloserine's been around for a very long time, and according to all the literature, L-cycloserine will inhibit an enzyme upstream of the synthesis of cycosine, but still ultimately inhibit cycosine. How much inhibition are we getting to get this very dramatic synergy, or this very dramatic increase in lifespan? Well, surprisingly, so these are animals, this is an untreated twitcher animal, these are twitcher animals treated with L-cycloserine. And what you can see is there's only about a 25% decrease in cycosine due to the L-cycloserine. So we don't have to completely inhibit the production of cycosine with a small molecule drug. A relatively small reduction leads to this very large response, which was surprising to us. We were actually hoping we'd get an 80 or 90% reduction, but all it took was a 25% reduction, which if you look at it a different way, could be a good thing. So then we have some unusual data. And if we have a good neuroanatomist in the crowd, if they can explain this to me, that would be great. We use the sciatic nerve as a surrogate for the peripheral neuropathy. And this shows the pathology, typical pathology in the sciatic nerve of a twitcher animal. Here's wild type. These are cross sections of sciatic nerve. You see all these really nice axons. You see they're wrapped in myelin. Everything looks happy in there. This is in a 36-day-old twitcher animal. This is a mess. There are fewer axons. The axons are smaller in diameter, and there's less myelin around these axons. In addition, there's an enormous amount of edema, and you get this infiltration of these macrophage-looking cells. Their sciatic nerves are a mess, okay? In the 36-day triple-treated animals, although the central nervous system seems to be relatively well treated, at least early on in the disease, there's essentially no effect of this triple combination treatment on the peripheral neuropathy, or at least the sciatic nerve. Again, you see fewer axons. These axons are smaller. There's less myelin. You still see this rather dramatic infiltration of inflammatory-like cells. Their sciatic nerves are a mess as well. And what's interesting, the animals that live longer, you get this really unusual histology, which we looked it up in a book, and the book said, this is onion bulb formation. And the book said that they believe this is due to repeated rounds of demyelination and remyelination. And you get these, what basically look like concentric rings around these axons. Now again, if you look at the axons, they're still small. There's very few of them. They're spaced quite far apart. And again, these are quite old animals, actually. These were out, it was a little bit of a range, but probably 300 days or older. I'm not a neuroanatomist. I don't understand what this means, but that's clearly not normal, nor is that. So I think, again, although we're doing a reasonable job of trying to treat the central nervous system, at least early in the disease, we're doing an abysmal job on the peripheral disease. And I think it's going to be one of the major challenges as we go forward with this. So this was all fine, and that paper was just published about a month ago in Journal of Neuroscience. Well, since then, we've initiated an additional study, and somebody tried to reject my NIH grant because we continued to use AAV5, even though we were trying to be good scientists and only change one parameter at a time. We finally decided that it's time to step up and use a newer version of AAV, and that would be AAV9. It's believed that AAV9 will cross the blood-brain barrier. It's also, if you inject it directly into the brain, it has a broader distribution and will transduce more cells. It is a better vector for CNS applications. So we did the identical experiment to what I just showed, but the only change was we were using a newer generation AAV vector. And there's supposed to be a slide there. There it is. So this study is still ongoing, and we have not done a lot of the analysis. We haven't done the Gal-C measurements, the Psyche-C measurements, that sort of thing. But I can show you the lifespan curve. Okay, and actually there's a bunch of curves on here for comparison purposes. This is untreated twitcher. These are animals that received the gene therapy plus L-cycloserine, so a dual treatment. You see, again, some modest increase in lifespan. I've already shown you these data. This is the lifespan from animals that received the gene therapy plus bone marrow transplant. Median lifespan is about 130 days. I just showed you these data as well. This is the data with AAV5, gene therapy, and L-cycloserine. Again, the median lifespan is about 300 days. These are the animals that received AAV9, bone marrow transplant, and L-cycloserine. And you see this increase in lifespan. The median lifespan in this group is about 400 days. So we added an additional 100 days onto the lifespan of these animals by changing AAV serotypes. Okay? Now, so this is good, and we're very excited about this. But there's a hidden story within these data, and that's here with all of these asterisks. Okay? These asterisks represent animals that died not from Krabbe disease, but from hepatocellular carcinoma. Okay? And in fact, this brown curve is the control group, wild-type animals that received the same regimen of therapy, AAV9, bone marrow transplant, L-cycloserine. And these are untreated wild-type animals. All these animals are dying of hepatocellular carcinoma. And in fact, all of these twitcher mice are dying from hepatocellular carcinoma. They're not dying of Krabbe disease at this point. These two probably did, and we don't understand why they died so early, clogged needle, who knows what. But these guys are all dying of hepatocellular carcinoma. And what's interesting is when these guys died, clinically, and I say clinically, behaviorally, these animals were absolutely perfect on the rotarod. They were also perfect on the wire hang test. Very simple test. You put your mouse on the wire, flip them over. Can they hang on? None of these animals here with AAV5 could successfully do the wire hang test. All of these animals could, right up until the day they died. Okay? And in fact, I haven't updated this curve in a while. There's 4 animals left in this study, 2 of which are litter mates. That's why there's only 3 little ticks there. These animals are now over 500 days of age. They can still perform the wire hang perfectly. They can still perform the rotarod perfectly. And we are keeping our fingers crossed that they don't develop hepatocellular carcinoma, although I think the odds are against them at this point. But again, the good news is we've dramatically extended their lifespan again, and they're not dying of Krabbe disease. Okay, they're dying of something else. It's a problem for the AAV field that needs to be addressed soon, I think, because this is not a unique observation. So we're excited about that. Again, one major question that we always try to ask, though, is these increases in lifespan are fine. We're very happy about that. But the real question in my mind is, you know, if we extend their lifespan, are we also improving their quality of life? I mean, if we tried to translate this into a child and we triple or quadruple their lifespan, but they're confined to a wheelchair, G-tube, respirator, have we really done anything meaningful? And that's not a question that I'm willing to stand here and argue about, but it's an important question. So, and again, we haven't analyzed all the data on these animals yet, but they say a picture says 1,000 words. This is a 30-day-old twitcher mouse. You can see the tremor. You can see the hind limb ataxia. This is a 450-day-old twitcher mouse. And it's pretty, they keep walking off the wildlife photography is not my specialty. But again, twitcher, treated twitcher, 450 days, 30 days. Okay, and to show that it's not just a difference between the mutant and the treated, there's a wild type in here now. So the same 450-day-old treated twitcher and a wild type litter, in fact, the mouse is litter mate. And I can tell you, this is the twitcher, and I can tell because it's got a little bit of kyphosis going on here. It's really very dramatic. And again, we're not just extending their lifespan. These animals really look very, very good, which is a real change for this disease. So what are the conclusions? So what we believe is by targeting multiple pathogenic mechanisms, we can dramatically increase the efficacy. The AAV-mediated gene therapy provides a persistent source of GAL-C activity and persistently keeps the cycosine low. It doesn't eliminate it, but it keeps it low. And the bone marrow transplant synergizes with AAV by decreasing both the disease and AAV-associated inflammation. And an additional synergy is observed when we add an additional treatment and do substrate reduction therapy. So what we think is happening just in this cartoon, we know that the AAV vectors that we use have a very strong neuronal tropism. So we think that we're transducing a lot of neurons in the brain. And then in this particular case, there's supraphysiologic levels of enzyme. And we believe through cross-correction, we can also correct some of adjacent cells, whether they be oligodendrocytes, astrocytes, microglia. We do get some cross-correction. The substrate reduction therapy decreases the synthesis of cycosine, which of course synergizes with the other therapies. And the bone marrow transplant, by some mechanism, is having this very significant immunomodulatory effect. And by targeting these various mechanisms, we're now starting to get really what I believe are meaningful increases in lifespan and improvements in clinical outcomes. So where are we going from here? The first thing that we're trying to do is develop safer AAV vectors. This hepatocellular carcinoma issue is a real problem. And again, there's good news and bad news. Our mice are living a very long time, but they're dying of the treatment-related problems. They're not dying of Krabbe at this point. And don't get me wrong, I'm not trying to say we've cured the disease. We have not. They still have disease. They're dramatically better. So we need to develop safer AAV vectors. We also need more specific substrate reduction therapy drugs. L-cycloserine acts on an enzyme actually upstream of ceramide synthesis. So we're not just decreasing the synthesis of cytosine, we're decreasing the synthesis of ceramide and all of the other molecules that trail off of ceramide. So what we really want to have is a galactosyl transferase inhibitor. Okay, it would be much more specific, presumably safer. We'd like to start using small molecule anti-inflammatories, which we've got some of those studies initiated now. And if I can blue sky for a minute, if we can effectively target the neuroinflammation, we may be able to get rid of the bone marrow transplant component of this regimen, which would be outstanding. We're also in collaboration with Ernesto Bongarzoni up in University of Illinois, Chicago, identifying new pathogenic mechanisms which we might be able to target and increase efficacy even further. And we're also collaborating with Charles Veit at the University of Pennsylvania to move some of these combinations into the canine model of Krabbe disease. And the obvious and first combination that we have actually on the ground now is CNS-directed gene therapy and bone marrow transplant in the canine model. I don't have any data to share yet, but those animals are being generated right now. And just to give credit where credit's due, the old MPS 7 data that I showed was done by Tom Daly and Anthony Frizella. Darshong Lin and Adarsh Reddy did the first combination treatment in the Twitcher mouse, the gene therapy plus bone marrow transplant. Jackie Hawkins did the study with the triple combination that was just published. And Yetta Lee is performing the experiments where she's now using AAV9 with these other combinations. And I get funded by a number of people, and there's other colleagues that have helped with this and other projects we have going. And with that, I'll stop, and if anybody has questions, I'd be happy to answer, whatever I want to do.
So obvious answer, I'm sure, but I'm not seeing it. What happens when you switch the sequence of AAV vector and then blood stem cell transplantation? What happens if you do it in opposite order?
So do the transplant first and then the gene therapy?
Yeah.
We haven't done that.
Okay. What's the rationale for not trying? Let me rephrase the question. Would there be reason to think that using, I know you're not using very high radiation, but I'm sure there's some degree of DNA damage and cytotoxicity going on intracranially. Would that improve transduction, do you think, of vector?
Well, actually, it's an interesting question. And the bone marrow transplant actually does increase transduction. There's a fairly large body of data out there showing that AAV is a replication defective virus, and it needs helper functions. What's interesting, though, the wild-type virus, adenovirus provides helper functions, herpes virus can provide helper functions, genotoxic agents can actually mobilize AAV, wild-type AAV. And what we've seen is that when we do AAV injections, and then we give them some radiation, we get higher levels of expression. Now, the question about switching them, we did the AAV first primarily because we thought it was, even though it sounds kind of gruesome, it's actually less traumatic than the bone marrow transplants. We wanted to do that first, and then the transplant. But again, to get the other part of your question, the ionizing radiation does enhance AAV expression. Now, so you go from 3 to 5-fold higher than normal levels to 4 to 6-fold higher than normal levels with the addition of the radiation. It's an interesting biological phenomenon. Yes?
Thank you for that incredible summary. It was really, really useful to hear. As you are aware more than most people, I would guess, of course, there's been a considerable pressure, politically and otherwise, to initiate newborn screening for Krabbe, with the only current therapy available being bone marrow transplantation. And I think from what you've shown, bone marrow transplant by itself, at least in mice, is not a particularly effective intervention strategy. Would you care to comment on what you see the potential direction for intervention might be for children who are already being identified, whether they wanted to or not, by newborn screening?
Well, I mean, in the short run, their only options are to either go untreated or to get a bone marrow transplant at this particular point. It's a tricky question because, obviously we'd like to see this stuff move forward. We think, if we can demonstrate in the canine model that we get at least similar results, in other words, we see some sort of synergy, we see these dramatic improvements, we'd like to see this sort of approach move to the clinic. Now, I've talked to the FDA about this, and the FDA is tough. They are very reluctant to see, even if we cured these mice, they would be very reluctant to allow us to move a triple therapy into the clinic without showing the effects of each one individually first. Now, we already know what bone marrow transplant does, so that's standard of care. But basically, we would be adding 2 additional therapies onto the current standard of care. The typical FDA style, they didn't say no, but the bar is pretty high, to be honest with you. So, but my strategy, and I don't know if it'll be effective or not, but my strategy is to generate enough data, irrefutable data in the mouse and in the dog to say, this is a horrible disease, bone marrow transplant doesn't do much, it's invariably fatal disease, no quality of life, let's move ahead with it. I have no idea if the FDA will buy it, but time will tell. And I think those decisions will be coming around within the next, say, 5 years. Some of the canine studies are on the ground right now. We're moving to look for better substrate reduction treatments, better viral vectors. We'll see, in 5 years, we'll see where it is.
I was glad to see you have some studies going on in a large animal model. I was wondering if you could tell us just a little bit more, like how much vector you put in and what route of administration you're using.
Yeah, so that's a good question. I don't remember the numbers off the top of my head, Scott, but the first question we had to address was the question that was raised a little bit earlier. What do we do first? I mean, this is a large animal model. Do we do bone marrow transplant first and then do the gene therapy, or do we do gene therapy first, or what? And it took some preliminary experiments to get that figured out. With the dog, we can give lethal conditioning. So with lethal conditioning, if you do that first and then you go in with the gene therapy a little bit later, these animals are immunosuppressed, you're putting in a virus, if there's even the slightest amount of contamination, it's probably going to end badly. So we did the pilot studies, and we're doing the CNS gene therapy first, allowing them to recover for about 2 weeks, and then doing the bone marrow transplant. But I forgot the exact dose. The route of delivery that we're doing right now, though, is intracranial, and we're putting in 8 burr holes, so 4 on each side, and we're delivering virus at 3 levels in each burr hole. Okay, so we're trying to give as much virus as possible, and so far the animals are tolerating it extremely well. So we'll see. I don't remember the dose off the top of my head, though. Yeah, puppies. So we're waiting for the animals. They're not weaned yet, but they are, I think, 3 weeks of age. So not neonates, real neonates like we do with the mice. Got to let them get a little bit older, but puppies for sure. Yeah, and they are pre-symptomatic at that point. So that's another important consideration. And that gets back to the question of newborn screening. We wanted to try to do the puppies again when they're pre-symptomatic to try to prevent the onset, because at least the data with bone marrow transplant would suggest that in Krabbe disease, once they're symptomatic, Doesn't matter what you do, you're not going to correct it. Now, we haven't tried our triple therapies yet, once the mice become symptomatic, but those are future experiments.
Do dogs get hepatocellular carcinoma too?
You know, that's never been reported yet outside of a mouse. And in fact, up until about 2 months ago, we were the lone voice in the woods about this hepatocellular carcinoma thing. But there's now at least 5 published papers where they show that if you do an IV injection of an AAV vector, and you let the animals live long enough, that's the other thing. You know, if you kill your animals at 6 months, you're never going to see hepatocellular carcinoma. It's a very protracted phenotype. It starts at about a year. But depending on the dose and depending on the serotype, you can get upwards of 75% penetrance that these mice will develop hepatocellular carcinoma. The good news is Charles Venditti published one of the more recent papers, and he showed that if you simply modify the promoter enhancer, you know, because we all think that if you just have a super duper promoter and, you know, a rocking enhancer, more is better. You know, typical American thinking here. And in fact, from an enzyme perspective, it probably is better. But Chuck changed the promoter. I think he actually eliminated the enhancer and used a weaker promoter and got almost no hepatocellular carcinoma. So it might actually be a relatively easy fix to avoid the hepatocellular carcinoma. But the other thing we don't know is whether this is a mouse-specific phenomenon. Nobody's seen it yet. It's clear it happens in mice at this point. But following intracranial injections, people haven't seen it. And we see it, though, because we actually see quite a bit of virus leak out following our viral injections. And we're giving radiation. So there's your first hit, there's your second hit. And we're seeing what will probably be about 100% penetrance of that phenotype. But I think we need to ask the hard questions on that, though. You know, is it a problem outside of the mouse? And if it is, you know, we don't want to give them a bad disease on top of a bad disease.
All right. Better wrap that up, I think, at this point. So, again, thanks, Mark, for
Thank you.
a beautiful presentation.